Don't stop. Don't stop.
Centrioles. AII cells capable of division, and even some which do not divide, contain a pair of structures called centrioles. With the light microscope, the two centrioles are seen as dots embedded in a region of dense cytoplasm which is called the centrosome. With the centrioles are seen to be short cylinders that lie at right angles to each other. When we examine a transverse section across a centriole, by M, it is seen to consist essentially of a series of microtubules arranged in a circle. There are 9 groups of tubules, each group consisting of 3 tubules. Centrioles play an important role in the formation of various cellular structures that are made up of microtubules. These include the mitotic spindles of dividing cells, cilia, flagella, and some projections of specialiæd cells, example the axial filaments of spermatoæa. It is of interest to note that cilia, flagella, and the tails of spermatoæa all have the 9+2 configuration of microtubules that are seen in a centriole. Projections from the cell surface. Many cells show projections from the cell surface. The various types of projections are described below. Cilia. These can be seen, with the light microscope, as minute hair-like projections from the free surfaces of some epithelial cells, Fig 1.26. In the living animal, cilia can be seen to be motile. Details of their structure, described below, can be made out only by M. A scanning M view is shown in Fig 1.27. A transverse section across the shaft of a cilium is shown in Fig 1.28. It has a striking similarity to the structure of a centriole, described above. There is a central pair of tubules that is surrounded by 9 pairs of tubules. The outer tubules are connected to the inner pair by radial structures, which are like the spokes of a wheel. Other projections pass outwards from the outer tubules. As the tubules of the shaft are traced towards the tip of the cilium, it is seen that one tubule of each outer pair ends short of the tip, so that near the tip each outer pair is represented by one tubule only. Just near the tip, only the central pair of tubules is seen. At the base of the cilium, one additional tubule is added to each outer pair, so that here the 9 outer groups of tubules have 3 tubules each, exactly as in the centriole. Microtubules in cilia are bound with proteins, dynein and nexin. Nexin holds the microtubules together. Dynein molecules are responsible for bending of tubules, and thereby for movement of cilia. Functional significance of cilia. The cilia lining an epithelial surface move in coordination with one another, the total effect being that like a wave. As a result, fluid, mucus, or small solid objects lying on the epithelium can be caused to move in a specific direction. Movements of cilia lining the respiratory epithelium help to move secretions in the trachea and bronchi towards the pharynx. Ciliary action helps in the movement of ova through the uterine tube, and of spermatozoa through the male genital tract. In some situations there are cilia-like structures that perform a sensory function. They may be non-motile, but can be bent by external influences. Such cilia present on the cells in the olfactory mucosa of the nose are called olfactory cilia. They are receptors for smell. Similar structures called kinocilia are present in some parts of the internal ear. In some regions there are hair-like projections called stereoci/ia. These are not cilia at all, but are large microvilli, see below. Abnormalities of cilia. Cilia can be abnormal in persons with genetic defects that interfere with synthesis of ciliary proteins. This leads to the immotile cilia syndrome. As secretions are not removed from respiratory passages, the patient has repeated and severe chest infections. Women affected by the syndrome may be sterile as movement of ova along the uterine tube is affected. Ciliary proteins are present in the tails of spermatozoa, and an affected male may be sterile because of interference with the motility of spermatozoa. Ciliary action is also necessary for normal development of tissues in embryonic life. Migration of cells during embryogenesis is dependent on ciliary action, and if the cilia are not motile, various congenital abnormalities can result. Flagella. These are somewhat larger processes having the same basic structure as cilia. In the human body, the best example of a flagellum is the tail of the spermatozoon. The movements of flagella are different from those of cilia. In a flagellum, movement starts at its base. The segment nearest the base bends in one direction. This is followed by bending of succeeding segments in opposite directions, so that a wave-like motion passes down the flagellum. When a spermatozoon is suspended in a fluid medium, this wave of movement propels the spermatozoon forwards, exactly in the way a snake moves forwards by a wavy movement of its body. Microvilli and basolateral folds. Microvilli are finger-like projections from the cell surface that can be seen by Fig 1.30. Each microvillus consists of an outer covering of plasma membrane and a cytoplasmic core in which there are numerous microfilaments, actin filaments. The filaments are continuous with actin filaments of the cell cortex. Numerous enzymes, and glycoproteins, concerned with absorption have been located in microvilli. Enzymes, and glycoproteins, concerned with absorption have been located in microvilli. With the light microscope, the free borders of epithelial cells lining the small intestine appear to be thickened. The thickening has striations perpendicular to the surface. This fry effect border of light microscopy has been shown by to be made up of long microvilli arranged parallel to one another. In some cells the microvilli are not arranged so regularly. With the light microscope, the microvilli of such cells give the appearance of a brush border. Microvilli greatly increase the surface area of the cell and are, therefore, seen most typically at sites of active absorption, example the intestine, and the proximal and distal convoluted tubules of the kidneys. Modified microvilli called stereocy/lot are seen on receptor cells in the internal ear, and on the epithelium of the epididymis. In some cells the cell membrane over the basal or lateral aspect of the cell shows deep folds, basolateral folds. Like microvilli, basolateral folds aid in adaptation to increase cell surface area. Basal folds are seen in renal tubular cells, and in cells lining the ducts of some glands. Lateral folds are seen in absorptive cells lining the gut.